All right. Well, today, this is going to be very open-ended today. So we don't have a presentation today. Really, what we want to get to is just have this as an open forum. I think that's going to be, the last ones have worked out really well, having somebody present, and would have, we'll do that in the future. But I think today, if we have some, this open forum where we can discuss ideas with everybody, on camera, if they're, feel comfortable with that. But then really using these, topics, challenges, and maybe potential solutions to draft, the next set of talks that will come from here. And so that's kind of where, sorry, I'm totally jumping ahead. I apologize.
Nope, I was just trying to get ahead with you. These are slides you all have, hopefully, if you're not new with us, have seen before. But that's just a quick introduction of the team. And here we are.
Yes. Excellent. So I would say, open up the floor to everyone. I think that would be really good. And if we could just kind of take notes on what people are saying, and if there's some themes that come out of that for what people really want to talk about in the future, I think that would be, that's what I hope to get out of it today. And then if nobody has anything to say, I would just lament about my own ineptitudes. So So I guess we had those little prompts. Maybe if we continue sharing those, just so we can see. So I forget what they were already.
I was also just going to quickly share, if it's okay, that Bina does have a ton of other events going on too. So the Lattice Lightsheet Group is here. We've got an early career working group that's kicking off. So if you work with people who are undergrad, graduate students, etc., please tell them about that. Share that. The group is meeting with the community to try and get information from the community. Our career series are wrapping up for the year. We've got our EOE still happening. We do have one last soft skills training, and then we've got our other 2 user groups happening, and Bina will also be at ASCB. So if you're attending ASCB, please check out our webpage on that. And hopefully that was a nice segue to give you all a moment to think about what your challenges and solutions and things are. But there you go. Sorry to interrupt, but also hope it was helpful.
Absolutely. Thank you. So I would maybe even also add one here, recent solutions. Are there any wins that anybody wants to talk about? It doesn't have to be doom and gloom either. Is there anything that was super exciting that you've recently have imaged and you'd like to take the time to brag a little bit? That's totally, I think, reasonable.
I guess I'll share something from my end. So I introduced myself in the chat. I'm the 3i Lattice product manager. So I'm from the commercial side. So I'm usually a little bit less exciting with the things I image. But we're working on a little white paper that we have with a user group at the University of Manchester that we imaged some adult C. elegans on the lattice. That was really fun. So they were looking at some single neurons, single proteins, motor protein complexes going along the different neurons in the C. elegans. I learned way too much C. elegans biology this summer. I'm a neutrophil and platelet biologist, but it was fun. It was fun to take like 3D printed sample holders and line the worms up in a nice little line. And even though they're huge, I'll admit they're bigger than the field of view in the lattice. They're millimeter long, but it was cool to do, subcellular resolution on a large organism, which is not something that people typically think about with the lattice, either from us or I see Courtney here too. But it's still possible. So something fun that we'll have up on the website soon. I'd love to show data, but if it was published, I could. Sure. I won't let someone scoop them unintentionally.
Yeah. Vanessa, please.
Yeah, just a point of, Andrew, you mentioned the sample holder and stuff like that. I don't know what may have been done by 3i and what might be open source, but Nikki, it occurred to me that that might be a good opportunity to talk a little bit about the tools database that the Microscopy DB is trying to move forward. So Nikki, I don't know if you want to take just a second to share about that, or I'm sharing my screen, but I can try and find some links in a moment.
Sure, no, I'm happy to pull up the links to that.
Thanks. But essentially, so Microscopy DB, if you all aren't aware of it, is a project that Bina has been involved in, but we're also involved with partners all across the globe. A lot of our other counterpart bioimaging organizations, and we have 3 databases currently, and then the tools database got launched at ELMI in June of this year. And that one is focused around like open source tools and hardware and software. And so it's just getting off the ground. We'd love to see more contributions to it. So I know Martin mentioned the new builders group. They will hopefully be contributing things to that. But yeah, it's an open resource, and hopefully the idea is to be able to disseminate tools with more of the community. So if people are building custom sample holders or different things for their light sheets, I can see that being of great interest to this group as well. So if you also have anything to contribute or know someone who's working on something that you want them to contribute it, the link should be available on the webpage that Nikki just shared to the ELMI page.
Yeah, thanks honestly for sharing that. I had heard of this, but didn't know the details of it. This collaboration we were doing with our users, they wanted to disseminate the sample holders because I can't even say it's my design. I helped them, but it's theirs. It's their work, their student. When they publish it, that's something I'll mention for them to do, is add it to, they were already going to think about adding the step file to the paper as like a supplemental thing, but adding it to a database sounds more useful for larger groups of people.
Yeah, the hope would be that then you can link to the publication or link to where the file is to 3D print it so that people can access it more readily and really get that technology disseminated.
That makes sense.
Thanks.
So just as a follow-up on that one, Andrew, was that like multiple C. elegans in one sample holder?
Yep. So like, I mean, I know you're familiar with the lattice sample holder, the upright Betzig style one.
Yeah, the spatula.
So yeah, we basically just printed grooves along. There's like 6 of them, maybe 7 of them, depending on the size. We tried a couple different sizes. I won't steal their thunder for when they publish this, but they're basically just ridges running along the cover slip that allowed us to settle the worms in. And then they have developed, along with some other C. elegans researchers, like a nice adhesion protocol to get them to actually stick, but remain, obviously alive, but also like semi-mobile, so that they're not just like completely glued down where they can't move at all, because that causes stress, but anesthetized and like kind of cradled in this little cocoon and gel. There was a fun polymer process the student worked on, but yeah, when it came to the sample, nice little ridges.
Awesome. That's super cool. You know, I was surprised. A lot of people, and you know, it seems like they still are using like the fixed tongs, like the forks on top of it. Is that what people are still, everybody's still using, like the glued or whatever springy things that are on top?
We use vacuum grease and just stick the cover slips on. The tongs failed over repeated use in our hands. And as the commercial side, we don't want people to be like, hey, your sample holder caused us to scratch the objectives. So we found a way around that, and we just got rid of the little springs and just used vacuum grease to stick it down. But more and more people really are printing them. We provide the file, even though we've tweaked it slightly, provide that file to any of our users who want it. I see Andy here from Case Western knows this too. And then they can print whatever they want. So you can micro pattern it for migration. You could do ridges for this. It's worked well, because if you're imaging, if you don't care about the epi path, you can.
Who does?
Yeah, I mean it's there. I like it for certain samples, but if you don't need it, well, it doesn't have to be glass. You can make it out of plastic or whatever you want.
Yeah, that's super cool. So for us, we got rid of the springs. Like,
Yep.
that was like the first thing that we did, but we ended up going with like just stainless steel and
Yeah, I could see that.
and slotting screws in it. And that seems to be a much easier process, because then we don't have to deal
Yeah.
with vacuum grease.
No, that's a good idea too. That's a good idea too.
So, how flat is it? The printed ones, are they really quite flat?
Less flat than glass, I'll be honest. But when you're imaging like the embryos, it doesn't matter in the end,
Sure.
which is why we get away with it. But yeah, it's definitely not as perfect. They depending on what resin you use, it shrinks weirdly. It doesn't always cure completely flat because it's so small. If you're doing some sort of pattern on it, it
Yeah,
doesn't cure it consistently across the surface always. We don't also have a super high-end 3D printer for this kind of stuff. It's meant for like larger prototyping parts. So when I make a tiny sample holder on it, it's
yeah.
But
Okay, so you're actually then plating the cells. If you were, so for like the embryos, you're putting them directly. That is, so it's a
Correct.
single use thing, and you just
single use thing for the cells. For the embryos, you can pry them off and reuse it a few times. It's until you gouge the plastic.
Okay,
So
interesting. That's super cool.
yeah, the cells, it's some sort of like suspended cells are what works really well for that, because then you're watching them attach and then migrate instead
Okay.
of just plating them on usually. But it technically works as long as the cells don't mind it. You can't really autoclave it because it'll melt the plastic. So you just gotta ethanol sterilize it and
Well, you can gamma radiate it too if you
oh,
had
that's true. That's true.
or UV.
Yep.
One of those two might.
Yeah, UV is what I've been doing.
Yeah. Awesome. That's super cool. Yeah, but the only time we printed one was somebody wanted to image some titanium cells on titanium, and so it was too thick to fit in on the normal sample holder, so then we just made one. But then like the bottom was so thinned out to get it to maintain the same shape that it was just like cantilevering over.
I could see that. I could see that. If anyone's going to be at ASCB, I'll make a note to bring one of those sample holders if they want to stop by and see it.
I'll be there. I'll be happy to stop by and see it.
Come on by and say hello.
Yes, that would be great.
We'll have the lattice there.
I think further shameless ASCB plug, Nikki and Bina colleagues will be hosting a sponsored session, but our corporate partners working group will also be having a roundtable discussion at the lunch on, was it Monday?
Yes, Monday at 1:15.
Yeah.
Roundtable.
So Andrew, if yourself or any other corporate colleagues want to join that discussion and are available, that would be great.
Yeah, can you say the time again? Sorry, I just
I'll
missed it.
swing by and see
Okay.
the sample holder, and I'll give you details.
Perfect. That's fine. Yeah, we'll have a few of us there.
Great. But yes, if there are any BINA members in the audience presenting that want to put their, drive some attention to your presentation, feel free to put it up, submit through our webpage. We have an events page for that. And then if 3i is doing any special workshops or anything like that, you can put those in the DB events page. So in the microscopy DB, there's an events page as well. So we're always happy to have you guys highlight workshops, or we find actually a lot of the online stuff does really well there, because that content goes to particularly the Africa bioimaging community are interested in the online free content. So they get a sort of specialized view of any of the webinars and things like that that go online. You can always submit that and find them on our page, which is here. Drop that in the chat. So this goes for any of you. If you have events you want to disseminate or jobs you want to disseminate, you know that we have this relationship with Microscopy DB where you put it in on the BINA site and it goes to a whole bunch of other partner networks as a result of just dropping it in one place. Anybody else got images they want to share or discussions they want to share around challenges they found recently? Would it be worth sort of having you guys share with the group what it is, you know, you're hoping to get from some of these meetings, and maybe if any of you are interested in presenting at them, because I know that we are always on the lookout for people to present.
Mhm.
Everybody's very shy this morning.
I know.
Oh dear, I'm actually driving people away by talking. We lost somebody. Sorry.
Asking for participation, Nikki. How dare you?
I know. I know.
Hello. Maybe I can ask a question.
The floor is yours.
Hi. Oh, well, thank you. This is my first time in the group. I'm Behzad Kajavi. I'm optical engineer at Rockefeller University. Just to introduce myself. I just wanted to know if there are builders here as well. I'm about to build a Lattice Lightsheet, and I wanted to see if you discuss such a thing, if we get a problem in optics, electronics, or anything along the way. Just wanted to see if this is part of the discussions as well or not.
I think that's totally, it has not been, but that does not mean that it cannot be. And so, yeah, if there's things that you would like to discuss, I think that's totally reasonable. I would say there is, what's his title? The chair of the new Bina Builders Working Group. Martin Fisher is here. So yes, there's at least one bona fide builder.
Right. That's more like a general thing, I guess. I thought maybe Lattice Lightsheet Group themselves, they have a separate part for this.
So I would say Martin built, right? Correct me if I'm wrong, Martin. You Lattice Lightsheet.
Yes.
There.
Correct.
So yeah.
Perfect. Okay.
But
So
there,
maybe to chime in a little bit. So
Yes,
the
please.
builders group, of course, so the Lattice Lightsheet will probably not be one of the focus devices, because it has a pretty good and well-established support already.
Mhm.
I mean,
Yes.
if you sign the license agreement with Genelia, you'll get access to all their myriad of files. You're going to be participating in the Lattice Lightsheet group, right, where you can post questions, and actually people will read it and answer it. So that's one of the examples where there is a good support already out there. The builders group will at least initially focus on other devices where that's just not existent yet. So if you build a Lattice Lightsheet from scratch, the user group will be your first line of attack.
Okay.
Watch the videos, right, which I'm sure you'll get the link to. Do all the files, print them out, look at what other people asked in that user group, and there's most of the questions that you're going to come across are going to be answered there. And of course, if not, you can post the questions. You pretty much know who the people are that built it. You can, if you know somebody who's recently built one, feel free to send them an email. I'm sure they wouldn't mind if you have a question, send an email to me. It's been a while since I've assembled one, but I'm pretty sure that the community is very, very open.
Very good. Well, thank you.
So you said you're just getting started building yours. Are you just getting all of the parts, or are you starting to build a line? Where are you at in the process?
Well, you know the long lead time items, right? So I have received most of the parts. I'm waiting for the 2 breadboards, custom breadboards from Newport. And then, yes, once they are built, sent, and then some custom parts that our precision, we have a section, the pit, we call it, and they are building the custom parts. It's a slow pace, but it is moving forward. So once I get those, I have the lasers, cameras, and objectives, all the rest almost. So I'll always start putting things together. But every setup has its own challenges, right? And this is going to be I guess the last version, because the last version of Lattice Lightsheet, because I was in Genelia and I talked to the team last year in December, I think. And yeah, so I got all the materials. So this is completely different from that video that was shared in 2016, I guess. So I have the AutoCAD file. So I'm looking at the components, well, optical components, and maybe some electronics as well, to see if I can. For aligning, I have less pain during the alignment and wiring and the rest.
Still upright?
Sorry?
It's still an upright configuration, correct?
Yes, it is. It is.
Yeah. Cool. Very
But
good.
then they have separated the beam structuring module and the laser combiner. So you have the laser combiner. It shoots up the laser to the top horizontal breadboard, that is beam structuring module, and then it is the upward microscopes that has the 2 objectives for illumination and detection, and the cameras. So yeah, it's pretty much organized, and I guess yeah, it's compact. So
Okay, cool.
Yeah.
Are you building this for a core, or are you building this for a specific?
yes, I am at the Bioimaging Resource Center at Rockefeller University. This is a core facility. So
Awesome.
a research group will be, yes, using it for their own research topics, whatever biology they are looking for. So
Excellent. Excellent.
Yep.
How many lasers are you going to put on it?
3. We
Just
are yeah, 3 lasers, yes, from because the questions here are mostly within those range. So 488, 561, and 642, right?
Right.
So they're looking for the fluorescence of those. So anyhow,
Awesome.
but you can always add the things. But I'm building the setup, so in the future, if there is any need to add more lasers, we can do it within the range that was. Yeah.
Awesome. Well, good luck getting started on that. And
Oh, thank you. Yeah, well, still waiting for
yeah.
the custom parts too. But anyway, yeah. Well, things are. It took for focus scanner, for example, it took around 10, 11 months. So.
Yeah. But it's not so there was like what I think the worst I've heard was resonant galvo. Somebody waited like 18 months for a Cambridge
Oh wow.
resonant galvo. I'm like, good Lord, that sounds terrible.
That's right. Yeah.
So.
I inherited some galvos, so I hope, yeah, I don't get any hiccups in the middle of, yeah. But then I was not, I was building the lab, so it wasn't like I was waiting for them. So the optics lab needed to be built. So
Awesome.
Just my quick comment on galvos. So yes, lead times are immense. However, I started to not order them from Cambridge anymore, but through Thor Labs, because what's actually in Thor Labs are the Cambridge scanners. And Thor Labs has them in stock, and if they are low on stock, they're going to get them first before we get them.
Sure.
yeah. Okay. I had well, thank you for that. Tor Labs had problems too. I guess there is no Galvo there right now on their website. Most of them are obsolete now. At least 10 months ago that I was looking. I had problems with one of their drivers, and it took them 6 months to fix something. I don't know. It's something disastrous is happening to the Galvos in the world, I guess. Yeah, it's like hopefully they have the Cambridge ones. Yeah.
I will also say the you know the Edmunds there, they sell the Scanner Max Galvos, which those are quite. I've been happy with that one thus far. So if you're needing something rather than
Nice. Would that be a problem with the software that they have for the Lattice Lightsheet? I don't know. Sometimes these things get into trouble. So if you get something that is not on the part list,
yeah, those are all driven with voltage though, right? So that shouldn't be an issue.
Yeah. Well, that's not a problem. I mean, how can I put it? They need a driver to recognize the board, right? So is the code that is written for the Lattice Lightsheet compatible with any type of Galvo that I get? Because obviously we need to have a driver so that the LabVIEW, a standalone software, can read it.
Mm-hmm.
I'm just afraid of that. I have had in the past some problems with these things, that the entire software is designed in such a way for certain things, and if you don't have that driver or whatever for the Galvos, then the entire thing just stop working.
Yeah.
So I don't know. Have you ever? Okay.
So I mean, ours was built in like 2017, following I think before the great Galvo shortage, and so yeah, I don't know, but they're just going to be driven by like an analog.
Analog output for the yes. Okay.
So
Good.
I would be shocked if they didn't.
Okay. There shouldn't be any problem. Okay.
I mean, I guess naively, I would say I would be surprised if they didn't work.
If they did, well, yeah, I sometimes get surprises sometimes from something that was be so obvious, but
yeah,
okay.
I don't know if others wanted to chime in and
your biggest challenge will probably be retrofitting the mounting of these things.
Okay. Well, thank you. Okay. Any suggestion how to be prepared for that moment?
I mean, if you really need to switch scanners, you have the CAD files, right? I mean, drop them in there, and you're going to have to make your custom adapter to make sure that it actually sits in the right place.
Mm-hmm. Right.
That's all. That's all there is to it.
Okay.
I guess there's more to it. I'm not sure about the new design. If you have the XY design, so two scanners that are in the same holder, of course, that distance you can modify, right? It's given by the scanner.
That's right. Yeah. Mm-hmm.
Depending on if that's different or the same, you're gonna have to do some modifications there as well. So I'm not sure about the new design. If is it two scanner separated in X and Y, or is it a combo X Y?
No, separated X and Y, and
okay, with some lens in between. Okay, so that makes it easier. Right?
I think it's adjustable. Yes,
right.
We can do that.
Okay.
Just okay.
Yeah, of course. Then you're gonna have to match the sizes, right? Make sure that your speed is okay. I mean, if you have jumbo mirrors, of course, they're going to be slower, right? If you have tiny mirrors, you might clip on them. So there's a trade-off there involved, right? Obviously.
Right.
Did you get it? You should have hopefully gotten the new and improved detection objective and excitation objective. Make sure you get that as your so we had to replace both of ours on our instrument, which just makes me not super happy. Because it was totaled about 15K extra that we had to replace both those objectives and get them repaired through no fault of our own. So make sure that the you know, they're it's basically the sealant on both of them around the nose piece went bad. And so that was one thing that was unexpected that happened to us.
Oh,
So
new challenges. Okay.
yeah,
Sure.
I mean, they're supposed to have been updated and the new version. So like they should be like glossy, the shrouds of each of them. So that's just like now my one word of caution to everybody is just like check it before the warranty goes out as well.
Right.
So
Well, thank you. Yeah, good point.
that's an unexpected. Ends up being a lot. Andrew, have your users experienced that much?
Yeah, Special Optics went through like 3 revs of that objective. One of our first ones, the coating failed and the front lens just dropped into the bath when they were loading a sample.
Okay.
So that was circa 2017, probably honestly same gen. But yeah, they've modified it extensively and have usually been fixing them on their dime, not on our dime, which is really nice. Because the minor effect is you get water inside the lens and you see lensing. And you're like, why does my single vessel look so poor?
Right.
You have an extra lens. You didn't know about. But yeah, so we haven't seen it in years.
Okay.
I imagine the fix you're talking about is the same fix we've seen.
So what we just had to replace was our detection objective.
We have not had issues with those.
Okay. And then there we had on the back side of the front lens a whole bunch of dye actually and stuff in there. So again, that same seal failed, same idea.
Yeah.
And actually, you know, we were thinking about it. It kind of makes sense because they were designed for this, not this. And so that was like, oh, yeah, I guess it's still surprising that the sealant failed, but it is not really being used to the design.
No, that makes sense. That's I'm gonna look out for that on the detection lens now.
Yeah.
But yeah, we haven't seen it.
Okay. Yeah.
Maybe we're lucky.
6-month ordeal getting that.
Yeah, we do not like getting those fixed.
Yeah, in fact, getting shipped back to Japan actually, and getting repaired that way. So, yay.
I always tell our users that when they break that kind of stuff, it goes back for spa treatment along with fixing because of how long it takes.
Yeah,
So they're pampered.
Yep, an extended vacation. So. One other thing that bath issues and sample issues that we've run into quite a bit is if we find if we don't clean the bath like just perfectly, something residual sticks around and just our cell health just is atrocious. And then if it's like, so I don't know if others have seen this as well, where it's like a very particular cleaning regimen that needs to be taken place. Yeah, so there's a curiosity on how others have solved this, because we now have a very stringent, like, if you don't do it this way, you're getting kicked out of the lab, basically. Okay, it's just our lab, so that's good. That's good. Well, any other thoughts from people? Any ideas for what you guys would like to talk about in the future, in future meetings? I think that's one thing that we haven't quite, we talked a little bit about building and things. And so I would say, even during the meetings, if there's like something here, feel free to, it's going to be a little less frequent, it's every other month. So it's probably not the best place to come and get your most pressing needs solved, but they're always welcome to bring them up for discussion. But if others have something like, oh, this would be a very interesting thing to present on, I think now would be a good time to discuss that, if you'd like.
While people are thinking on other topics, just wanted to quickly flag that I'm guessing nobody wants to meet January 2nd, which would be the next 2 months away, first Tuesday of the month.
Yeah, I would say let's push it back to the 6th. That's a bit.
Let's go to February. But great, just wanted to confirm that with everyone. And so yes, I will update this slide and update our website accordingly after this meeting. But yes, any topics that you guys want to see discussed at a future meeting, or I know some folks have suggested future speakers that we can reach out to. If you have someone else too that you'd like to see speak at one of these, we're open to suggestions, and we're happy to keep a running list of people we can contact who are available at our dates and times.
Yeah. All right. Well, I think everybody has. I think this has been fun for me still. So thank you all for engaging. And I think for the next missed what? Oh, missed the meeting. I see you're just arriving. Let's see, you're just arriving. I was only wrapping up because nobody wanted to talk, and I didn't think. But if you have, so there's an open forum. So if you want to hop in and please. Keep this going. We do have 15 minutes. And so there's 15 minutes of open time. If people, again, want to talk, let's talk. Otherwise, I'm not going to take any more of your time. Is all. Going once. All right. Well, I guess I'll take everybody's silence is ready to be off. So we'll put this on pause until next year. Be the what was it, the 6th of February. We'll be there, and we'll be back to regularly scheduled presentations, I suppose. So thank you all, and we will see you in February.